Hello, Mrs. Warnock here. I wanted to take this opportunity to go through your DNA structure notes with you. I know we've been talking a lot about DNA structure during class. You guys did the race for the Nobel Peace Prize, which kind of allowed you to go through the data that's gone into how the structure of DNA was discovered, and so I wanted to kind of take that information and put it into more of a formal notes situation. So, DNA structure. The first essential question, and you can fill this in on your note packet right where it says essential question, is what is the chemical and physical structure of the DNA molecule? And you guys already have quite a bit of information in this. So, along that topic, the first question is what is DNA? So DNA stands for deoxyribonucleic acid. And you'll remember from doing our DNA structure puzzle that deoxy means that the sugar that makes up that sugar phosphate backbone of DNA, the one in DNA is missing an oxygen. And that's why we call it deoxyribo for deoxyribose, which is the name of the sugar. It's called nucleic because, as we found in Oswald Avery's study, DNA is found in the nucleus. And Oswald Avery's study also determined that DNA has 65% acidic properties, or in other words, DNA is slightly acidic. So here we've just got a little picture of, the nucleus, which is where the DNA is found. And the nucleus has all the DNA kind of neatly wrapped up into these little structures. And a nucleus is obviously an organelle inside a cell. So each of your body's trillion cells, with the exception of red blood cells, contains about 2 meters of DNA. That's in every single cell. And that DNA is coiled into a chromosome inside the nucleus. So, 2 meters of DNA, that's a ton of DNA. And when you think about you have 2 meters in every single cell, how is that DNA able to be packaged and stored? If you were to actually take all of the DNA that you have in every one of your cells and you were to unravel it, take it all out of its chromosomes and stretch it out, it would actually go from Earth to the sun hundreds of times, not once, not twice, but literally hundreds of times. So you have a ton of DNA inside every single one of your cells. So DNA is a molecule. And it's kind of like, if you think about it, like a cookbook or like a book that has all of the blueprints that tell all of your cells what to do. So the DNA is kind of like the cookbook. And what are the parts of the DNA molecule? DNA is in the shape of a double helix. So that's kind of like a twisted ladder. And here, if you look at the screw kind of shows you how the DNA is twisted. And the ladder, if you think back, is kind of like how you had the sugar phosphate sides of the ladder, and then the rungs were those nitrogenous bases, adenine, thymine, cytosine, and guanine. And if you remember, those guys were held together by some bonds called hydrogen bonds. So what are the parts of the DNA molecule? The building blocks of the molecule are called nucleotides. And here we have a diagram of a nucleotide. And you guys have already done some work with looking at the structure of a nucleotide. Up here, if you look at the left-hand corner, you've got your phosphate group, which is the thing that attaches, to a sugar. Here you've got your little house-shaped deoxyribose sugar. And over here to the right, you're going to have your nitrogenous base. And if you remember, we talked about there could be, multiple rings. So these little hexagon shapes are kind of like the rings. And if you have 2 rings, that's called a purine. And if you have 1 ring, that's called a pyrimidine. So again, what are the parts of the DNA molecule? Just to kind of recap on that, nucleotides are composed of, number 1, a phosphate group, which would be this guy right here. Number 2, in the middle, we'd have our deoxyribose sugar molecule. And up here in the right, we'd have one of our 4 different nitrogenous bases. And those guys are called adenine, thymine, guanine, or they could also be cytosine. So those phosphate and sugar groups are kind of like the backbone of the DNA molecule. So here you've got phosphate, sugar, and you'll notice on the other side that the phosphates and sugars are actually upside down. So one side of the ladder is called 5 prime. So what that means, if you look at it, I'm trying to point with my finger like you can see. If you look at it, you can see up here the 5 prime end is the end that's closest to like the point of the sugar, the point of the house, the top of the house. And the 3 prime end, we call it, is next to the flat end or like the base of the house. So those guys are flip-flopped, and that's actually known as being anti-parallel. How is the double helix structured? So in between those backbones, the sugar phosphates, we have the nitrogenous bases. You'll notice that C is held to G with 3 hydrogen bonds right here. And then T and A go together, and those guys are held together with 2 hydrogen bonds. So if we look at like the sequence of C's, T's, A's, G's, T's, that's actually the genetic code. And that is what makes you, not only is it what makes you human or makes you your species, because every living thing has DNA, but it's what makes you different from your brother or your sister or your friend. It's what makes, different hair color and eye color and all different kinds of things, different personalities even. So how do the nitrogenous bases pair together? Adenine always binds to thymine with 2 hydrogen bonds, and cytosine always pairs with guanine. And you're going to see that cytosine and guanine have 3 hydrogen bonds. And what is a hydrogen bond? It's simply a bond between 2 hydrogen atoms. And we learned about hydrogen earlier in the year when we talked about chemistry. So that's just called, when 2 hydrogen atoms connect, that's just called a hydrogen bond. So in order for the base pairs to fit, we did a little thing with this when we were doing that puzzle for the Nobel Prize. There was a study that they tried to figure out, well, which goes with which? Do purines go with pyrimidines? Do pyrimidines go with pyrimidines? And we figured out that the best fit, like the Goldilocks and 3 bears for purines and pyrimidines, is that a purine, which is this double ring, remember these little hexagons are called rings, so this double ring bonds to this single ring, and the double bonded to the single is called a purine to a pyrimidine, and those guys are the best fit for the model that was developed by Rosalind Franklin, James Watson, and Francis Crick to discuss the structure, of DNA. So we're going to just talk about some easy ways to remember how the bases pair together. So the 2 circle-shaped letters pair together, and the 2 stick-shaped letters pair together. So C is like circle-shaped, G is like circle-shaped, A and T. Another way to remember this is that 2 angry tigers, so adenine and thymine, pair with 2 hydrogen bonds, and 3 calm giraffes, so cytosine and guanine, pair together with 3 hydrogen bonds. Another way to remember, how many hydrogen bonds there are and which pairs with which. So then trying to remember, well, which is a purine and which is a pyrimidine, you can remember pure as gold. So adenine and guanine are purines, pure as gold. AG stands for gold. Cut the pie, cytosine and thymine are pyrimidines. So just a little trick to remember those guys. So you don't have to answer these questions for homework. We're going to do the sum it up questions when we start class the next time we meet, but you do have to fill in all the spaces in your note packet. All right, that's all. Thanks. Bye.